So I'm Xavier Heiligenstein, a postdoc at Institut Curie for the past 5 years now, and I've been recruited after my PhD to establish and develop methods of correlative light and electron microscopy for Institut Curie, and also within the development at that time of the France bioimaging infrastructure. The idea was to make a method that would be user-friendly and usable by most scientists from Institut Curie, but also from everywhere in France, and being able to propose them a way to come, do the experiment, and go back with easy results to get. Correlative microscopy is actually a scientific approach to combine images from different modalities. Whether they are coming from light microscopy or electron microscopy, then it turns into correlative light and electron microscopy, CLEM. And the idea is to be able to follow something by live cell imaging, and at a given time fix the sample to prepare it by electron microscopy, and then combine these two images into one single image. The combination of these two images can be done by informatics and allow us to track the dynamic information into the ultrastructure of the electron microscope. To achieve that, we first need to have high-resolution light microscopy, and then being able to fix the sample to transfer it to the electron microscope. Light microscopy brings us specificity and dynamic information about what's happening in the cell. On the other hand, electron microscopy will give us ultrastructure information and contextual information at much higher resolution, but everything has to stay static. It's not dynamic anymore, it's fixed. And so the whole thing around correlative light and electron microscopy is to be able to go from light microscopy to electron microscopy without killing the sample or disturbing it too much so that we do not retrieve it well. For that, we developed a new method centered around the CryoCapCell, which is a technology I invented during my PhD, that allows us to follow a structure which is located according to landmarks, and at a given time take the whole sample and transfer it into a high-pressure freezer that will vitrify the specimen before preparing it for electron microscopy. What we figured is that we are not going fast enough between the live cell imaging and high-pressure freezing. So together with CryoCapCell, which a company I co-founded a few years ago, we developed a new high-pressure freezer which is attached directly to a light microscope. So we load the sample all together, and at a given time when we see the event of interest, we press the button and the sample is transferred for high-pressure freezing. Once it's high-pressure frozen, we can prepare it for electron microscopy. And using the landmarks of the CryoCapCell, we can retrieve the sample in the electron microscope to correlate the images. We also figured that we have troubles in correlating the two images and evaluate the precision of the correlation. So what we did for that is we developed a software which is called CLEM, together with Perrine Polgilotto from Institut Curie, and now she's at the Nantes University. We developed this software to easily correlate the two images together and to have an evaluation of the statistical error of the correlation. So now we have an entire workflow that allows us to go from live cell imaging to high-pressure freezing and image correlation using both the CryoCapCell, the high-pressure freezer, and CLEM, the software for correlation. So the application of correlative microscopy in general would be studies of molecular mechanism. A year ago, we published a paper on the block 1 complex protein, which is a complex involved in the formation of the endosomes and the tubulation of the endosomes. And when block 1 is mutated, the endosomes are not forming properly anymore, resulting in a neurodegenerative illness, which is called Hermansky-Pudlak syndrome. So we've been able, using correlative microscopy, to figure the localization of the block 1 and the function of block 1 protein regarding the ultrastructure. Also, some colleagues have developed correlative light and electron microscopy approaches at the EMBL in Heidelberg, where they've been studying the progression of a single tumorigenic cell through the blood vessels, and to be able to track the entrance of these tumorigenic cells into the tissue. So they've been following it by a 2-photon microscopy, and at the given time of interest they've been fixing the samples, preparing it for micro-CT analysis to have a large view of the block, and then retrieve it by electron microscopy to understand exactly how the cell, the cancerogenic cell, manages to go through the blood vessels and to enter the tissue. So correlative microscopy has the ability to observe things from different levels, bringing both the dynamic information and merging it with the structural information into one single information. That gives us generally a better understanding of what is happening, and it also allows us to find the needle in the haystack. The single cell penetrating the tissue is something that happens very rarely, and by electron microscopy we couldn't find it. So rather track it by light microscopy, and once the event is happening, fix it and go and have a look at the electron microscopy level.